Hello everyone, my name is Ragad Rifai. Today we're going to talk about the recombinant DNA technology. The recombinant DNA technology is a technique mainly used to change the phenotype of an organism when a genetically alerted vector is introduced and integrated into the genome of the organism. So basically, this process involves the introduction of a foreign piece of DNA structure into the genome which contains our gene of interest. This gene, which is introduced, is the recombinant gene, and the technique is called the recombinant DNA technology. There are multiple steps, tools, and other specific procedures followed in the recombinant DNA technology, which is used for producing the artificial DNA to generate the desired product, which we will understand more in details in the next slides. So what is the recombinant DNA technology? It's a technology used for producing artificial DNA through the combination of different genetic materials from different sources, which is referred as recombinant DNA technology. Inserting the desired gene into the genome of the host is not as easy as it sounds. It involves the selection of the desired gene for administration into the host, followed by selection of the perfect vector with which the gene has to be integrated and recombinant DNA formed. Thus, the recombinant DNA has to be introduced into the host and at last has to be maintained into the host and carried forward to the off. The tools of recombinant DNA technology. The enzymes, which include the restriction enzymes, help to cut the polymerases, synthesize, ligase, and bind. The restriction enzymes used in recombinant DNA technology play a major role in determining the location at which the desired gene is inserted into the vector genome. There are two types, namely endonucleases and exonucleases. The endonucleases cut within the DNA strand, whereas the exonucleases remove the nucleotides from the ends of the strands. The restriction endonucleases are sequence specific, which are usually palindrome sequences and cut the DNA at specific points. The desired genes and the vectors are cut by the same restriction enzymes to obtain the complementary sticky notes, thus making the work of the ligase easy to bind the desired gene to the vector. So what are the vectors? The vectors help in carrying the integrating and desired gene. These are very important part of the tools of the recombinant DNA technology. Also, the vectors are made up of an origin replication. This is a sequence of nucleotide from where the replication starts. A selectable maker consists genes which show the resistance to certain antibiotics. Also, we have the host organism into which the recombinant DNA is introduced. The host is the ultimate tool of recombinant DNA technology, which takes in the vector engineered with the desired DNA with the help of the enzymes. There are a number of ways in which these recombinant DNAs are inserted into the host, namely micro injection, biolistics or gene gun, alternate cooling and heating use of calcium ions.
Hello, my name is Ragad Nassar. I will define the process, application of recombinant DNA and cloning DNA. The complete process of recombinant DNA technology includes multiple steps, maintained in a specific sequence to generate the desired product. First step is isolation of genetic material. The first and the initial step in recombinant DNA technology is to isolate the desired DNA in its pure form, like free from other macromolecules. Step 2 is cutting the gene at the recognition sites. The restriction enzyme play a major role in determining the location at which the desired gene is inserted into the vector genome. These reactions are called restriction enzyme digestion. Step 3 is amplifying the gene copies through polymerase chain reaction. It is a process to amplify a single copy of DNA into thousands to millions of copies once the proper gene of interest has been cut using the restriction enzyme. Step 4 is ligation of DNA molecules. In this step of ligation, joining of these two pieces, a cut fragment of DNA and the vector together with the help of enzyme DNA ligase. Step 5 is insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into a recipient host cell. This process is termed as transformation. Once after the insertion of the recombinant DNA into the host cell, it gets multiplied and is expressed in the form of the manufactured protein under optimal conditions. As mentioned in tools of recombinant DNA technology, there are various ways in which this can be achieved. The effectively transformed cells or organisms carry forward the recombinant gene to the offspring. Now the application of recombinant DNA technology. The first one is DNA technology is also used to detect the presence of HIV in a person. Also, gene therapy, it is used as an attempt to correct the gene defects with give rise to hereditary disease. The third one is the clinical diagnosis, which called ELISA, is an example where the application of recombinant. And the last one is the field of medicines. Recombinant DNA technology is used for the production of insulin. At last, DNA cloning. A clone is a cluster of individual entities or cells that are descended from one progenitor. Clones are genetically identical as the cell simply replicates, producing identical daughter cell every time. Scientists are able to generate multiple copies of a single fragment of DNA, a gene which can be used to create identical copies constituting a DNA clone. DNA cloning takes place through the insertion of DNA fragment into a tiny DNA molecule. These molecules is made to replicate within the living cell, of instance a bacterium. The tiny replicating molecule is known as the carrier of the DNA vector. And thank you for listening.